Now, as a quick recap to what we had covered in the first two webinars, there were concepts of databasing and knowledge management. We highlighted how physical chemical predictors can be used to help with designing experiments. And then we highlighted some chromatography tools, both free tools that are available on the web, as well as some of the tools that are available for purchase. And some of the highlights were on chromatographic simulations, method translation, as well as column characterization and selection. So at this point, I'd like to turn things over to Kareem, and he's going to give you the presentation on webinar 3.
Hi, everyone. Thank you for taking time out of your busy days to join us for this webinar. I'd like to start off by introducing you to my friend Charlie, who apparently has a headache. Charlie is an expert chromatographer who works at a major pharmaceutical company. He and his team are responsible for generating stability indicating methods for the company's drug products that are going into development. As you likely know, stability indicating methods are analytical methods with baseline resolution of the API, its process impurities, and degradation products with no coevolution. That's quite a challenging task. Stability indicating methods are very important as they're typically used hundreds to thousands of times to identify materials that are out of specification or out of trend for quality control to ensure efficacy, safety, and quality. Moreover, if a coeluting component is discovered in a stability indicating method after it's been established, that method needs to be redeveloped and revalidated. This has significant consequences from a cost and resource perspective, as well as influencing the drug's time to market. Although Charlie and his coworkers are using some of the software that was mentioned earlier to assist them with their method development, it's not quite adequate for their needs. So as you can see from the slide, the nature of stability indicating method development requires analysts to look at a number of different samples simultaneously. So they could be looking at their API control, they could be looking at degradation samples, so acidic, basic, peroxide degradation, and they may have reference standards that they want to evaluate at the same time. For specificity, they want to analyze columns, so typically they'll select a number of orthogonal columns to get the best separations possible. They'll evaluate buffers and different solvents. Once initial methods have been identified, then they would take those initial methods and do further optimization by tweaking either the gradient, the temperature, or other factors in order to reduce the runtime or to improve the separation. Now, as you can see, this would result in a huge amount of analytical data, and at the end of the day, they have to make a decision based on that information. If they're doing chromatographic modeling, then peak tracking becomes important. So typically, if there's a large project with many components and many experiments, that peak tracking could take a significant amount of time. It's routine work, but it's monotonous, and it bites a lot of your time where you could be spending it doing analytical chemistry. Some of our customers, such as Charlie, said that they would keep track of all these experiments in an Excel spreadsheet. At ACDLabs, we refer to that as dead data because there's no convenient way that you can go from a spreadsheet back to the analytical data to interrogate what you were observing. Okay. Other things that are important to Charlie and his coworkers is robustness. They want to make sure that any slight variation in conditions, such as temperature or gradient, don't dramatically impact the performance of the method. And finally, Charlie's boss attended a seminar recently and heard about quality by design. So another task that was put on Charlie's plate is they have to follow quality by design principles to ensure that the methods that they're delivering are of a certain quality. Okay. While Charlie is a hypothetical persona, we have encountered countless individuals with the same challenges, and I expect that many of the attendees of this webinar can relate. In fact, we worked very closely with a number of individuals like Charlie at different companies, including Janssen Pharmaceuticals and Eli Lilly & Co., to develop and iteratively refine an automated software solution that directly addresses these challenges. So I'm going to describe at a very high level the 5 steps that are part of this software and automated solution. We'll do it at a high level, and then my plan is to go back over all 5 steps and describe them in a little bit more detail. So the first stage of this process is to define a quality by design strategy with critical quality attributes and success factors. This governs the 5 steps that you're seeing in front of you. The first step is direct instrument control. The second step is automated peak processing and peak tracking. This is integrated with a data management system where all the information that was generated from the acquired data resides and allows you to make quick decisions based on observing the live analytical data and chromatographic information. This is tightly integrated with chromatographic simulations. As you've seen from the previous webinar, we have a product called LC Simulator which does chromatographic simulations. This capability has been tightly knitted into the solution that you're seeing today. And finally, once you've achieved the desired separation, we have capabilities that allow you to clearly communicate those results to your colleagues or within the industry. Okay, so let's start off with step number 1. The software has a very easy to use graphical interface, and it allows you to configure your system or to set up the configuration of your system. So you can specify what instrument you're working with. You can specify what columns are being used. Now, in this case, as I mentioned, you'll use a series of orthogonal columns and thermostats to control the temperatures. You'll specify what samples you're going to be measuring, and in this case, you would have your API, you may have your degradation samples, you may have internal standards or standards as well. You can also specify what solvents and buffers are set up on your instrument. And whether you're detecting using PDA or mass spec detection, or both. So once you've set up the software and defined what your system looks like, it will use that information to build a strategy, as you're seeing on the right-hand side. Now, just at a high level, this strategy that was recommended involves column and buffer screening, selection of the best one, followed by optimization of gradient and temperature. You can specify certain quality attributes within the strategy so you know when you've achieved your desired goals. Now, assume here that you've set up the system and you've specified 8 columns and 2 buffers. How would the system behave after that? So we can take a look. On the left-hand side is where you set up the software, and your strategy is determined that you're going to be doing 8 columns and 2 buffers. Well, that results in a sequence of 16 injections. Now that's only if you're analyzing 1 sample. If you're analyzing 2 samples, then it would be 32 injections that would be built in the sequence. That sequence would be communicated to your CDS, and then the CDS in turn would communicate with the instrument, and would result in acquisition of the data. Once the data is acquired, that information comes back and is stored within the CDS. And then finally, a copy of that information is brought into Autochrome for next steps. So we have instrument control with Waters and Power, as well as Agilent ChemStation for those chromatographic data systems that we don't currently support. There's a virtual offline mode that can be used. It has all the same capabilities, except you don't get the instrument control that you do with the other two systems. The advantages of instrument control is your injections are actually built on a quality by design strategy with critical quality attributes defined upfront. This results in dramatically improved efficiency. There's other steps that are included here, where there will be a purge and equilibration step involved after every injection to ensure the highest quality results. And finally, and I think very importantly, this avoids transcription errors. As you've seen, when you're doing 16, 32, or many more injections, there's a chance that you can make errors, and they can be pretty costly. Okay, so once the injections are completed and brought back into the software interface, we have the capability to do automated peak tracking. So as I'm representing here, only with 2 chromatograms, it's a pretty simple task to track peaks if you're only looking at 1 or 2 peaks. However, if you have areas where there's coelution, that can be a little bit of a challenge. And if you have areas where you have peaks that are switching positions, as shown here. 1 second, I'll just use my highlighter. So where you see peak switching position or new peaks that are appearing that weren't present previously, that becomes very challenging. And we're only looking at 2 chromatograms right now. Imagine doing that for 16, 32, or more injections. So we have the capability to do peak tracking by using PDA data. And what's utilized is a solvent-subtracted UV spectrum to do the matching. If you have LCMS data, then we have a deconvolution, chemometric deconvolution algorithm that will deconvolute your signals and automatically identify the molecular ions that are observed for each component. If you use both MS and UV, then what the software will do is allow you to reconcile that information, and this results in a much more global way of being able to do peak tracking. The final advantage here is because of the way the system works, automated peak tracking will actually identify and detect co-eluting peaks when it would be very difficult to do that manually. Okay, so once the peak tracking is done, what I'm showing you here is a table that's built in the software. Once the peak tracking is done, it's displayed in the grid as displayed in front of you. On the left-hand side are the 16 experiments that we've asked the software to acquire. This is essentially the 8-column 2 buffer grid that we discussed earlier. And then what you can see on the right-hand side is the molecular weights for all the components that were present. And you see the retention times for all those components in this grid under the different conditions. Finally, the software will calculate a number of things such as It calculates the total number of peaks that are observed. Obviously, when you're doing stability indicating method development, you want to find the conditions that give you the most number of peaks. You don't want anything co-eluting, so fewer the number of peaks that are observed means that there's some co-elution. And you can see that in this grid where there's holes, and that means that that peak that was present in a previous injection is no longer visible, so it can't be identified. Okay. Now, this is a graphical representation. I want to show the depth of this table. So it's more than just a simple Excel-like spreadsheet. It contains the same information that somebody would have if they were to put all this information into a spreadsheet. However, when you click on a row, you can see the chromatographic information behind that row under those conditions. Now, this is unique to ACDLabs. What you're seeing is a composite chromatogram. And what that means, this is essentially a synthesized chromatogram, and it's under one set of conditions showing peaks from both MS and UV detectors as well as components from different subsamples. For example, this chromatogram contains peaks that were observed for the API, acidic degradation, which is the red colored chromatogram, as well as process impurities, which are shown in orange. In addition, if I were to click on this peak, I can readily see the mass spectral information and the UV information associated with that peak. If I know the structure, I can add it, and now this propagates through the entire sequence of injections that was done. And all the metadata and instrument parameters get stored and is readily accessible. So just to give you a little bit more of an idea, we're looking at the API peak right now, but say I was interested in knowing what this tiny little peak was, and I wanted to identify what the mass spectrum and the UV spectrum is for that. Simply clicking on that peak or selecting it at the table displays the mass spec and the UV spectrum along with the parameters and metadata for that peak. So as you can see, this is essentially a multi-dimensional collection of information that stores all the mass spectral information and UV information for this injection. Okay. The next step, step number 4, is around optimization. With our strategy, we had selected gradient and temperature optimization. And what I'm going to describe on the left-hand side is a two-dimensional graph, which is the gradient time versus the temperature. And in the background, what the software has done is it's acquired data to be able to successfully model this design space. So the white dots that you're looking at are actual experimental chromatograms. And these experimental chromatograms are used to model the rest of the surface. Areas where it's blue, there is a coelution of critical pair of peaks. So that's not the ideal locations where you want to be for chromatography. And the reddish-orange color specifies that there is a good separation between all peaks. And so you get a bird's eye view of your separation conditions with respect to gradient, time, and temperature. Another representation of the same information is on the right-hand side. So you can clearly see with this where the areas of highest probability of separation would be. Now, if you were doing method development in a linear fashion, so say we started at a particular temperature and we went up the gradient, you could find that, okay, so now the separation's starting to improve, and you could optimize the temperature and end up in an area where there's a local maximum. However, the way the software works by sampling the entire design space, it gives you the capability of identifying where the global maximum is simply by having a few experimental data points collected and then modeling the rest. By sampling the entire design space also, you can identify areas of robustness. So if we look at the left-hand side, you can see that there's areas that have considerable robustness. And what I mean by that is you have enough room or enough latitude in the gradient or temperature that you're not going to dramatically impact the separation. And this is very critical for when you're transferring from one laboratory to another, or if you're in the same laboratory but transferring the method from one instrument to another, because there's going to be minor changes and you want your method to be as robust as possible. Taking a look on the right-hand side, while it's important to have a large platform or good robustness, it's equally important to have a knowledge of where the edges of failure are. You don't want to be stepping too far over on the right or the left here, but it's good to know how far over you can move. Okay, so once the modeling is complete, that suggested experiment can be run by the software and brought back into the software interface, and you can evaluate how well the modeled chromatogram fits with the experimental data that you've just acquired. Taking a look at the screen right now, it looks like a relatively reasonable fit. And if this fits my critical quality attributes, then I could be finished. If in fact the model was different from what my experimental results are, I can put that additional experiment back into the software and have the model redefined or reconfigured and suggest next experiments based on the refined model. Okay. The last step, step number 5, is communication of the results. As you can see, we can generate really nice reports within the software, and they can include many of the elements that you've seen. So you can have a strategy included in your results. You can have metadata included, tables of experiments that were run, results from that. You can include resolution maps, like a 1D resolution map if you're optimizing one factor, or as you've seen, you can have a 2-dimensional resolution map with many factors. In addition to simply just reporting the results, ACDLabs has a database platform which allows you to database chromatograms, spectral data, method information, metadata, as well as structures associated with this. So we have organizations that are using our databasing platform for chromatographic data as well as spectral data to share within their organizations, either locally or broadly within different sites in different countries. Okay, so in summary, we have covered a software package that involves automation. The steps that were included were a quality by design strategy with critical quality attributes that allows the user to define their success criteria based on their available instruments and inventory. This strategy governs the 5 steps. So the first step is instrument control. And as I mentioned, we have direct instrument control with Agilent ChemStation and Waters and Power. Once the data is acquired, it's easily processed automatically in the background, or peak tracked. Once the data is processed, it goes into a data management interface. This data management interface includes structure information, chromatographic information, spectral data, as well as metadata and tables, all in one place to enable you to make decisions easily. All of this is tightly integrated with chromatographic simulation software. And once you've acquired an ideal method, then you can communicate your results to your colleagues or within the company. Okay. I was going to go into a brief software demonstration just to show you some of the elements that I'm describing here. The parts that I was going to go over was at the beginning how somebody would start off by designing a quality by design strategy within the software. And then I'm going to show you an interface where the project is nearly complete. So what we'll see is the data management interface after peak tracking has been done and after instrument control has acquired the data. And then I'll show you how the chromatographic simulation interacts with the data management interface. So I'm going to stop sharing for a minute while I get ready. And please hold for a few seconds. Okay. Brent, can I ask you, are you seeing the right screen there?
Yes, we are.
Okay. So I've started off the software and we're configuring the instrument parameters. Essentially, it allows you to specify what instrument you're working with. So I'll just hit next. You get to define the columns that are available. This is a very easy to use graphical user interface where you can simply drag and drop from one pane to another, or you can make adjustments if you wish. So double clicking in here allows you to make any changes that you need to. You can specify thermostats as well. If I go next, this is my solvent delivery system. And as I said earlier, the software comes with predefined templates. You can always modify or add your own, but we have quite an extensive list of templates already available with the software as it comes. I'll just hit next, and now we're looking at the detectors. This is set up for diode array and MSD detector, and you can change it from series to parallel. Clicking next, and here's my samples. So I've specified that I'm going to use a tray and I'm going to run a sample and my acidic degradation sample. I can add a new one if I wish simply by right-clicking and adding and defining. Okay, and so this is created. I can drag and drop it into the tray and say okay, and now we have 3 samples that are going to be evaluated. Hitting finish, and you're brought back to the software screen. I'm going to open the task, and now here's where the strategy can be set up. So as I said, the software defines or suggests what strategy you could use. Within here, you can decide on what specifics you want included, as well as your critical quality attributes. So if there's certain settings that you require, you can change them. Just click OK here, and now what the software will do is it'll use that information and it'll generate a list of experiments. One very handy thing that you'll see the software does is there's an assistant that guides you through the entire process. So a user might define this. They have a chance to make any changes before it's executed, and then hit execute experiments, and that would trigger the information being sent to the chromatographic data system, and in turn going to the instrument. Okay, and I'll also open up a finished project and walk you through some of the elements of that. So what you're looking at is, let me just switch to the strategy so you can see what was done. So this is a project that's at the very last stage. So what's happened is buffer screening, column screening, and gradient and temperature optimization. I can go back and look at what happened with the buffer screening. So we ran 3 experiments at different pHs, and the software automatically determined that pH 2 seemed like it was the best one. And you can see why, because you have the drug product and you have various impurities that were observed, and there are coevolution in the last 2. So the first experiment at pH 2 gives the best results as far as the number of peaks. We can go to the next stage where it was column screening, and it took the best one from the previous screen and applied it to column screening, and these 2 were evaluated. The Hypersil Gold was chosen as the best one with the best resolution. And then we can take you to the final one where there was a gradient and temperature optimization that was conducted as per the screen. Just show you that if I go into the chromatogram tab, I can very easily select my experiment and display so the chromatographic, the chromatogram for that specific injection. Okay. We can also display, so what's happened here is the experimental data was brought in the background, a model was created, and what the software did was it calculates or simulates chromatograms based on the experimental. If we turn on both the predicted and experimental chromatograms, what we can see here is there's very good correspondence between the experimental and the simulated chromatograms. As you can see, there's black and red, and they're pretty much indistinguishable in this example. And you can evaluate that with every single run that was done. Finally, the last experiment, this is the simulation of what the software deems to be the best optimization experiment, and it's essentially waiting for you to execute the experiment and bring it in to confirm that it matches up with the prediction. Okay. I know there's a lot more within the software that we could demonstrate, but if you're interested in seeing more, please feel free to contact ACDLabs, and we'd be happy to set up a meeting where we can show you more and discuss this in more detail.